So let's just jump to the slide that talks about that.
At the highest levels of motor control, you have the motor control systems in the brain. So you have the motor cortex, and the motor cortex will be communicating with the sensory cortex because the sensory cortex is providing information about where the body is, where the limbs are you standing upright, are you sitting down, are you laying flat. All of that comes from information from the sensory cortex. And the association cortex is, of course, one of the things that helps to tie information together. So there's the highest levels of control are where you will plan your movements. You will have information from all your systems and you'll put together your plan. The plan might be to throw a ball, the plan could be to type the letter G on the keyboard, the plan might be to do a backflip. Whatever it is, this is where everything comes together for the plan. There's an area just anterior to the motor cortex called the premotor cortex, and it also contributes as a higher center. So you think of this as areas in the cerebral cortex, the higher centers of motor control. In the middle levels of motor control, you have areas which are specialized to communicate with the different muscles to have them activated at a particular pattern. So if I want to move my arm in a circular motion, there is a pattern of muscle activation that has to occur. And the middle layers of motor control will control that pattern. And so the higher centers will make the plan, I'm going to move my arm in a circular motion, but then they'll communicate with the middle layers of motor control, which will provide, oh, well, then you're going to need to activate this muscle, and then this muscle, and then this muscle, and this is the right sequence to get that motion to occur. That's what happens in the middle layers. Also in the middle layers is where if you want to hold something steady, the middle layers are paying attention to any drift. So if you get a little off course, the middle layers help correct your course so that you can still hold something steady. This is important because people who have some motor control disorders like Parkinson's disease have problems in the middle layers of motor control. And the reason that they have a tremor is because they no longer get that programmatic feedback that says, no, you wanted to hold your hand steady. That becomes a complication when you're trying to eat soup with a spoon, right? Lifting it to your mouth, if you can't hold the spoon steady, you may spill the soup. So the middle layers of motor control are essential for giving you that control of fine motor control, the feedback that comes back from the sensory system that tells the middle motor control areas, oh, you need to make a little correction here, right? So middle layers provide the programs for muscle actions. The lower levels of motor control, that's where the work is done. There you have the motor neurons that connect with the muscles. And so the higher levels and the middle levels will plan motions, will plan the pattern of activation for the motor neuron circuitry, but the lower levels is where the action is. That's where a command is sent from a cell body in the spinal cord out to a muscle in the periphery. And if the lower motor neurons fire an action potential, muscles will move, unless that nerve-muscle connection has been disrupted in some way. Now you can see here they also include in this particular diagram that some of the local circuits in the brainstem and spinal cord are in this lowest level, and that's where you can get feedback from sensory systems for reflexes. You have a sensory system that detects a change in your environment which requires a rapid change in your body position. That's a local circuit. And a local circuit can cause changes in muscle movement that don't require the middle levels or the upper levels. And so when you step on something sharp or you touch something hot on the stove, you get a faster response because you have a local circuit. Sensory system that connects directly to the motor system, and the motor system that commands the muscles. I think that covers it. Yeah. So we need all three layers. There's a little division of labor that goes on so that you can have some areas that are specialized for the big picture issues and others that are focused on more specific details. And this diagram gives you more of an atlas of where things are. So the cerebral centers, the motor control centers, and the sensory cortex, motor control cortex, sensory cortex, will be here on both hemispheres. These structures are deep in the brain, and they include some of the middle control centers. So you have basal nuclei and some brainstem motor centers that we would consider part of the middle control. And then those cell bodies that are in the spinal cord connect to axons that then directly innervate the muscle. I see the thalamus here is a region that's dense in cell bodies, but it's within the area that is of the cerebral cortex that normally has white matter. So it's kind of a concentration of nuclei that can then contribute to control of muscles in the middle centers. The cerebellum is a specialized area that relates to control of muscle movements in the middle layers. And so you should think of cortical areas as being upper centers. These nuclei that are within the white matter and the specialized regions of the cerebellum and in the brainstem as middle layers, and then the lower levels just as spinal cords, neurons, and the muscles that they innervate. And this just shows the integration of contact of information at the lower levels. You have sensory receptors that you find in the periphery that communicate with the motor neurons that are at that level. Those sensory neurons can also send, and do also send, information to the higher centers. They ascend through the spinal column. But they can influence the activation of a motor neuron that controls a local muscle. Now, this information that goes directly to the motor neuron does not necessarily guarantee action of the motor neuron, but it is part of what the motor neuron receives. And just like Dr. Kierstead described to you, a cell body, a neuron cell body, can get multiple inputs from multiple sources, and then decisions are made. The decisions tend to be the integration of the synaptic inputs. So what are these decisions? Well, you get a hyperpolarizing signal from one neuron, you get a hypopolarizing signal from another neuron, right? Hyper says you should fire an action potential. Hypo says maybe not right now. You're depressed in your ability to fire an action potential. And it's the combination of the stimulatory inputs and the inhibitory inputs that then determine whether or not an action potential gets formed as those positive and negative signals come to the cell body. You have to have a large enough depolarization at the axon hillock or the beginning of the axon that's just as it leaves the cell body to depolarize the voltage-gated sodium channels in that vicinity before you get an action potential. And that's what's going on right here. This descending motor track is sending its inputs into that motor neuron cell body. The sensory neuron is communicating to that same cell body, and it's the combination of those inputs that determines whether or not an action potential is fired by this particular motor neuron. And that becomes something that we think about more when we talk about some of the complex reflex actions that we talked about during this lecture. I'm going to jump ahead.
And the main reason I want to jump ahead is because I've had I just looked today and there was another comment on the forum about muscle spindles and their action, so I want to talk about that. Yeah.
Is there kind of situation where the nerve is being innervated by both a motor signal and a sensory signal only occur in the spinal column?
No. So the question was, does this situation where a nerve is getting input from a sensory neuron and some sort of upper center only occur in the spinal column? So in the central nervous system, as part of our general communication pathways, most neurons have multiple inputs on their cell body. I mean, if we're talking about motor control, yeah, that's where the final decision is made, is at the motor neuron cell body in the spinal cord, if we're talking about motor control. But throughout the totality of the nervous system, the cell bodies of neurons have synapses with multiple other nerves. And so they are receiving information from more than one source all the time. And whether or not that cell that is on the receiving end then sends an action potential will always depend on the sum of the inputs that it receives. So whether you're talking about, if we could be talking about a neuron that's in the motor cortex in the brain. It's receiving sensory information, it's receiving information from the association cortex and the cerebellum and the premotor cortex, and eventually the sum of those inputs cause whatever neuron is at the heart of this to then either remain quiet or to send an action potential. And that's the way the nervous system circuits work, is that multiple inputs into a cell body, the cell body weighs them all, and the weight of that determines whether or not an action potential is then fired. And that sends information to another network of cells, and then they have to go through the same process as their cell bodies. Does that make sense? Okay. So I want to just touch on this because there's some aspects of it that I think sometimes people get confused about. So the muscle spindle is this kind of specialized hybrid cell, and the ends of the muscle spindle are very similar to normal muscle cells. They have myofibrils in them, and they're contractile, they can shorten. But the center doesn't have that kind of structure, and the center of the cell is wrapped by the nerve endings of a sensory neuron. And so, the best way to describe it is that the terminals of that sensory neuron are likely mechanically gated, because when the spindle is stretched or when the spindle is shortened, it changes the firing pattern. And so there's a physical change in the environment that causes the firing of this sensory neuron. And the purpose of the muscle spindle is to determine how stretched or how contracted is this muscle, and that information is constantly being fed back to the central nervous system. Constantly. These neurons never shut up. They're always sending signals. And the spindles are responsible for length changes, and the Golgi tendon organ is responsible for force changes that occur on the tendon near the joint. So we talk about this muscle spindle reflex, and I want to just walk you through it again. If you use a reflex hammer to tap on the patellar tendon, and we're tapping below the kneecap. You tap below the kneecap, that pushes in right here, below the kneecap, and by pushing in there, it causes a stretch of the part of the patellar tendon that's above the kneecap. And the stretch of that patellar tendon above the kneecap causes a stretch of the muscle and the muscle spindle, and it changes the firing pattern of the sensory afferent. Now, because this change in position, this stretch signal, is essentially a signal that's somewhat unanticipated by our nervous system, then the body circuitry interprets it as something that needs correction. Didn't think it shouldn't happen, so maybe we should fix it. And if this muscle was stretched, let's contract it. And so the sensory afferent connects through the dorsal horn, through the ganglion and the dorsal horn, into the spinal cord, and it is sending its information about that stretch event to the brain and to the motor neuron that connects to this quadriceps muscle. And it's sending an excitatory signal to the quadriceps muscle. And if you send and if that signal that is excitatory goes to this muscle, the quadriceps will contract. And when this quadriceps contracts, it pulls the tendon in the opposite direction. It contracts the muscle, it pulls, and then the foot will kick out because of that contraction. Now, to make sure that that happens, it communicates to this inhibitory interneuron. So this is another nerve cell in the spinal cord. And when this cell body is excited, it sends a signal to its nerve terminal, but the neurotransmitter that it releases is inhibitory on the cell that it synapses with. And it's synapsing on the motor neuron that connects to what's called a hamstring muscle, which is on the backside of the thigh. And so it makes it less likely that the hamstring muscle will also contract. Because if the hamstring muscle contracts at the same time as the quadriceps muscle, then you get a balance of forces on the kneecap and the leg doesn't move. But once the reflex says that initial movement that occurred because of the tap of the hammer, that shouldn't have happened, so you want your foot to move. So you inhibit this muscle, you activate this muscle, your foot goes from this position to this position. And it's a ability of the sensory neuron to both stimulate the what you might call, this is sometimes called the agonist muscle. The agonist would be the one that acts, and to inhibit the antagonist, the one that would work against the agonist. And so it's inhibiting the hamstring muscle. So that's basically the spindle reflex. The spindle reflex is a stretch on this side, on the lower side of the patellar tendon, that stretches this tendon above the patellar tendon, which stretches the muscle, stretches the spindle, and then sends the signal to the spinal column that immediately feeds back at the same level to cause a contraction there. Yes?
So are the lower and higher levels receiving the signal like near simultaneously?
So
very close to the same time. The major difference would be that this, just because of the geometry, that the lower center of response is quicker than the higher, because if you send a signal to the higher centers, then it has to process it and then send the signal back down. That's going to take a little bit more time.
So what's the point of it even going to the higher center?
You want the brain to be aware of what's going on.
Just like awareness?
It's awareness, and occasionally the higher center may decide to override.
Okay.
Right? The higher center may say, no, don't do that. So for this, I often like to think about pain sensors, especially say with respect to handling something hot. And I think it's the same example that your book you may use, or the critical thinking exercises uses. You pick up something hot. It's got your dinner on it. You're really hungry, but the plate's really hot. Now, do you drop the plate, or do you take your time to set it down? Well, if you want to eat it and not off the floor, you take your time to set it down. That's the higher centers kicking in saying, no, don't drop that. Even though at the lower level, the initial impulse is, that's too hot, let it go. Right? Yes?
Can you explain how the alpha-beta coactivation works in this?
I can do that, yes. So, thank you for that question. It was a good question. I like that one. So, when you stretch a muscle, the speed of signaling for a afferent connected to a muscle spindle speeds up. And when you contract the muscle, it slows down. So you stretch it, it speeds up, and when you contract it, so speeding up, this is the action potentials coming from the afferent. When you contract it again at this side, it slows down. That's the key of alpha-beta coactivation. That's a key bit of information because or alpha-gamma coactivation. In alpha-gamma coactivation, the issue is as the muscle shortens, that signal gets slow. And as the muscle shortens again, it gets slower, and it gets slower and slower. And eventually, if the signal is coming at too slow of a rate, it doesn't provide very much information. So, in order to make the signaling rate worth listening to, still provide information, what happens is the spindle has started to fire slowly because of the contraction of what's called the extrafusal muscle, the regular muscle cells, and the contraction has caused them to fire slower and slower. But it gets to a certain point and you activate the gamma motor neurons, and here's a gamma motor neuron going to the contractile ends of the muscle spindle. And if you contract this end and you contract this end, then you stretch the middle. It's as if I had a rubber band between my hands and I stretch the rubber band. That's what's happening to the spindle when both ends contract, is that it gets stretched. And that small stretch that occurs, the small stretch that occurs here essentially resets the signaling rate for the spindle so that it can continue to fire, rather than slowing down, it can continue to fire at a more rapid rate that contains more information. And it can continue to respond to the shortening of the muscle.
In this case, would you say that duration and amplitude are important for?
No. So the question was, are duration and amplitude important? In alpha-gamma coactivation, you would have an alpha motor neuron stimulating the regular muscle cells. And if it's a steady contraction like we normally have, it's firing at a relatively high rate. The gamma motor neuron's rate has slowed, and the activation of the gamma motor neuron to stretch the muscle spindle allows it to increase its rate a bit, to increase the rate of firing of that afferent. It's the rate that changes. There's nothing with amplitude that changes.
And the duration of the contraction will be determined by the length of time that the alpha motor neuron continues to contract the bigger cells, the full muscle cells. But there's no amplitude change of signal. Remember, we're talking about action potentials. Once they're fired, they're always the same size.
Can you just say again which alpha versus gamma is?
So alpha, so this is, when you think about a muscle in the body, the main muscle cells are innervated by alpha motor neurons. And it's just the terminal ends of the muscle spindle that are innervated by gamma motor neurons. So gamma motor neurons only to muscle spindles, and only at the ends of the muscle spindles, which are contractile. Alpha motor neurons, the bulk of the muscle cells, will be innervated by alpha motor neurons. The only time you have to worry about gamma is when you're thinking about the muscle spindle.
So
Yes.
then, does the alpha neurons act on?
Alpha motor neurons synapse on normal, regular muscle
Right.
cells, not on sensory systems like spindles.
So then, how does the gamma know to send the signal to strength?
That has to do with the sensory feedback. So you've got this feedback. Don't know why it's not responding. You've got this feedback system that's sending information from the sensory afferent to the spinal cord and then to the brain. And so the brain knows that the muscle is shortening. And when it reaches a limit where the signal is getting too slow, it activates the gamma system. So it sends a signal down to the cell body of the gamma motor neuron and then activates that system so that it will change the sensitivity of the spindle so that it can still get useful information. So it's a combination, so that sensory information that's always coming in about how long or how short this muscle is, there's an internal indicator that says, okay, the muscle is at this point and it's still shortening, we need to activate the gamma system so we can still get useful information. Okay, yes.
I think one day that we finished a little too late and we didn't get through all the slides. Can we go through the cycle of the skeletal muscle? Like, acetylcholine stuff.
Okay. All right, so the question was about, well, the question was about more than one thing. If you want to talk about excitation, contraction, coupling with EC coupling, we covered that in class pretty thoroughly, but we can talk about that pretty quickly as well.
We just meant like the summary part of the skeletal muscle. It's slide 11.
This one?
Yeah.
Okay, so this is a, summary of excitation, contraction, coupling. What's excitation? What is it?
on the slide.
Potential?
Excuse me?
Action potential?
Right, so the muscle has to fire an action potential. And it fires an action potential because it received neurotransmitter from the motor neuron. So there was an action potential in the nerve, and then it caused neurotransmitter release at the nerve terminal, diffused across the cleft and bound to the acetylcholine receptor. The acetylcholine receptor is a the acetylcholine receptor is a ligand-gated or ligand-gated, depending on whether you're me or Dr. Kierstead. If she says ligand, I say everything. She's Canadian. It's a ion channel that opens because it binds a signaling molecule. The kinds of molecules that pass through this channel are positively charged. But it doesn't care which way they go or how big they are. And so sodium can flow in and potassium can flow out and calcium can flow in. It turns out that more sodium comes in than potassium going out. And that means you get a local depolarization, which is called an end plate potential. Not yet an action potential. You only get an action potential if you open a voltage-gated channel, a voltage-gated sodium channel. So the response of the acetylcholine receptor is to have a local depolarization, which is a graded potential, which will spread from that site. As it spreads from that site, and this cartoon does not show it very well, it will encounter the voltage-gated sodium channels. And the depolarization that occurs because of the end plate potential will cause the opening of the voltage-gated sodium channels, which is the upshoot of the action potential. And action potentials are membrane phenomena. So this will be a change in the polarization of the membrane, and it will spread from the end plate to the end of the muscle cell. And since the end plate tends to be in the center of the muscle cell, you actually get action potentials traveling in both directions along the length of a muscle cell at the same time. A signal that starts in the middle and then goes to the end of the muscle cell at the same time. As it travels along the muscle cell, as that action potential travels along the muscle cell membrane, it will encounter these holes in the membrane that lead to channels that go through the muscle cell that are called transverse tubules. In the transverse tubular network, it will encounter the dihydropyridine receptor, which is a voltage-gated channel. It's a voltage-gated calcium channel. And so the dihydropyridine receptor will respond to the action potential as that wave of depolarization passes by, and it will move. Normally, this movement opens a pore that allows calcium to go from the extracellular environment to the intracellular environment. But so little calcium comes in through the dihydropyridine receptors that you find in skeletal muscle that nobody ever worries about it. It's as if it doesn't happen. What's important in skeletal muscle is that the dihydropyridine receptor is very, very close to the ryanodine receptor on the sarcoplasmic reticulum membrane. And the movement of the dihydropyridine receptor will cause movement and opening of the ryanodine receptor so that calcium can go from the high concentration inside the sarcoplasmic reticulum into the cytoplasm where the concentration is low. And calcium floods out of the ryanodine receptors into the sarcoplasmic space and starts to diffuse through the cell. And because the transverse tubules and the sarcoplasmic reticulum are essentially wrapped around the myofibrils in very close contact, the diffusion doesn't have to go very far. Before that calcium will encounter the thick and thin filaments, and on the thin filament, it will bind to troponin, and troponin is connected to tropomyosin, and the calcium binding to troponin will release the tropomyosin from its spot on top of the myosin binding sites, which are shown here as little black dots on the thin filament. With those sites exposed on the thin filament, the myosin, which has already bound ATP and hydrolyzed ATP,
can bind to actin and change its conformation, releasing energy and releasing the products of ATP hydrolysis. So it releases phosphate and it releases ATP as it makes a conformational change, and the myosin will pull on the actin-thin filaments to cause the movement or the force buildup that is contraction in a muscle cell. And it will continue to cycle and do that for as long as the calcium concentration remains high. For normal muscle movements, because you don't just have one nerve cell action potential, but a series of nerve cell action potentials, this means that the calcium remains high for quite a while, as long as that movement is supposed to occur. And you get continuous cycling of the cross bridges of the myosin during that process. However, when the nerve action potentials stop, then the muscle action potentials stop, which means that the dihydropyridine receptor then will block the ryanodine receptor, and so no more calcium can come out of the sarcoplasmic reticulum. During this whole process, there are calcium pumps in the sarcoplasmic reticulum membrane, and they've been trying to reacquire the calcium during the whole process. Now that the calcium release has stopped, the calcium uptake can reduce the cytoplasmic calcium concentration, and they pump the calcium back into the sarcoplasmic reticulum, which means that the calcium concentration in the cytoplasm eventually gets so low that it releases from the troponin, and the troponin tropomyosin complex then blocks those myosin binding sites on the thin filament. Yes?
So when the action potentials stop coming and then like there's less calcium, that's when ATP starts to bind?
When the calcium stops, you said, you have to repeat that for me.
When like the concentration of calcium starts getting lower, that's when the ATP starts to bind too.
ATP binds to what?
Is that, I thought you said earlier, is that the one that goes to myosin?
Right, okay. Let's talk about that. Myosin binds ATP as soon as it loses ADP, all the time. So during this process where myosin is generating tension or causing shortening of a muscle cell, it binds ATP, it hydrolyzes ATP, it changes its shape as it does that hydrolysis event. Then when it can bind actin, it releases ADP and phosphate, it reverses that conformational change, which transmits the energy to the thin filament and causes it to either move or causes tension to build up. But as soon as it lets go of ADP, it can bind another ATP.
So that's what keeps it moving?
That's what keeps it going. So that ATP binding hydrolysis release is going on and on and on and on and on as long as the calcium is present. When the calcium concentration gets low, myosin that has finished its power stroke will bind an ATP and hydrolyze it, but since it can't bind to actin, it stops. It basically stops. And it can't move forward with release of ADP and phosphate without binding to actin again, at least not for a very long time. Eventually, if the muscle was relaxed for a very, very long time, it would let go of the ADP and phosphate. And it would bind another ATP and start that process again. But for the most part, it holds it for a very long time, unless it binds actin again. Yes?
Does that mean that the myosin is always in like the cocked position when
Yes.
it's not contracting?
Yes, that's outstanding. So is myosin always in its cocked position? Almost always. Almost always. Myosin is primed and ready to go. Almost always. The only time it would not be is if the ATP concentration got very, very low, and that would mean the cell was sick. Anyway. So myosin is almost, so that when the signal comes, when the calcium gets there, myosin's ready to go. I'll go back to the contraction cycle in just a second. We can talk about that. But yes, that's exactly it. As soon as the calcium comes, the myosin's ready to go. So we block the myosin binding sites on actin. The myosin no longer can bind there and make force. The muscle relaxes. That's the end of the cycle. And so the excitation contraction cycle starts with the nerve stimulating the muscle to fire an action potential, the muscle action potential triggering calcium release into the cytoplasm, calcium release in the cytoplasm binding to troponin on the thin filament, which removes the troponin tropomyosin from a blocking position where they prevent the actin-myosin interaction. That means that myosin and actin can interact cyclically to keep generating force for as long as the calcium concentration is high. If the sarcoplasmic reticulum stops releasing calcium, then these calcium pumps, which also require ATP, can pump calcium against a concentration gradient into the lumen of the sarcoplasmic reticulum to store that calcium for the next contraction. I have asked this question before. I don't think it's on tomorrow's test. But there are 3 uses for ATP in the muscle contraction cycle. Number 1, the one that everyone thinks about, is it's used by myosin to generate ATP, or to use the energy of hydrolysis to make force. Number 2 is that the ATP is required to pump the calcium against the concentration gradient to get back into sarcoplasmic reticulum. Can you guess what the third one is? It's the hard one. The one that people don't think about. You remember when, well, some of you were here at the very beginning. I showed a little movie and I said the muscle was in rigor mortis. If at the end of the power stroke there isn't any ATP around, the myosin remains tightly bound to actin and doesn't let go. And so at the end of every power stroke, you absolutely require an ATP to detach myosin from actin so it can then go through the cycle again. So 2 of the roles of myosin are because of the ATPase action, either of myosin itself or of the calcium ATPase. The third role that ATP plays is that when myosin binds tightly to ATP, it can no longer bind tightly to actin, and so that allows myosin to let go. And especially if you have a shortening muscle, you want myosin to let go so the filaments can continue to slide and shorten the sarcomere. So the 3 major roles in a muscle cell have to do with the ATP that's required for the force generation, the ATP that's required for the calcium pumping, and the ATP that's required to release myosin from actin at the end of the power stroke. Now, if we wanted to get more technical, we could say, well, you also need ATP in the nerve terminal. You know, you need ATP for the sodium potassium ATPase. We can go into other uses of ATP. But I'm talking about just the contractile mechanism, those 3 main uses of ATP. So let's talk about this cycle of myosin using ATP. So myosin is an enzyme, we call it an ATPase because it splits ATP to ADP and phosphate. So it binds ATP and it splits it to ADP and phosphate. When it does that, it changes its shape as it makes that chemical bond break. And we interpret that as that's myosin storing the energy of hydrolysis. And myosin in a relaxed muscle is primarily like this. It has ADP and phosphate bound to it, and it is not tightly binding to actin. I started to say this in class, I don't know if I finished the thought. Myosin can bind ATP tightly, it can bind ADP and phosphate tightly, or it can bind actin tightly, but it can't bind both the nucleotide and actin tightly at the same time. So in this state, with ADP and phosphate bound, if the cross bridges were blocked from interacting with actin, it would just sit there and wait for the calcium to come. In this cartoon, the calcium's already there, and so the sites are open. But in a relaxed muscle, you either have myosin with ATP bound or ADP and phosphate bound, and most of it has ADP and phosphate bound. Over 80%, maybe over 90%. I've actually measured that years ago when I was a graduate student. How much of myosin was in the ATP state and how much was in the ADP phosphate state.
So if it's in the state where ATP's hydrolyzed,
Yeah.
it will bind to the actin as soon as it.
As soon as the actin is available, it binds to it.
Okay.
Yeah. That also brings up a point that's not really in your textbook. We talk about myosin binding to actin, and we talk about it being detached from actin, but we don't really talk about how it goes in that transition from being detached to attached. Basically, it's thermal motion. Is that myosin is sitting in this sea of water, and it's constantly moving around. And one way I've thought of it is that myosin's constantly bumping into the actin-thin filament, and it's asking the question, does the calcium come? Does the calcium come? Does the calcium come? And when the calcium hasn't come, it can't find its binding site. But as soon as the calcium is there, it can find its binding site and it can go through its contraction cycle. Yes?
If most of the myosin heads are already primed before contraction actually, or before the actin's actually available, how come they don't all like attach and detach at the same time?
Because their binding to actin is essentially due to that thermal random motion. They're not attracted to actin by some magnetic force as soon as the tropomyosin moves out of the way. Actually, if they get close enough, there are some charged particles, charged surfaces that help pull them together. But their attachment to actin is somewhat dependent on, were they close to actin when the tropomyosin moved out of the way, or were they far from actin when the tropomyosin moved out of the way? So they all don't bind together because they're not all sitting in exactly the same position during the rest state, but they're all sort of independently moving around. And when the binding sites become available, some of them will be able to bind very quickly, and others will take a little bit more time. It's a good question, but it's one of the reasons why it's not like a crew team that says, okay, all the oars are in the water and then we're going to stroke, and then we're going to stroke, and then we're going to stroke. It's not like that. It's somewhat independent. People have scratched their heads about that for many years. Why doesn't it do that? So you sit here in the relaxed state, ready to be activated. Once you get the activation, if the calcium's on the troponin, myosin then can bind to the thin filament, and it will release the phosphate, it will release the ADP. So it releases phosphate as power stroke starts. It starts to move the actin thin filament. That's why we have multiple arrows to represent that it's trying to move the actin thin filament. And then we'll lose the ADP. When the ADP releases from the nucleotide binding site on myosin, then it's available for the next ATP. And ATP can bind, but when the ATP binds, the affinity of myosin for actin is decreased, and then the myosin will rapidly hydrolyze that ATP to ADP and phosphate. And the reduction in the tightness of the binding between this state where the ADP is on and this state where the ATP is on is what helps to facilitate the filament sliding, because otherwise a myosin that was tightly bound at the end of the power stroke that stayed on would be acting like a brake. It would be interfering with cross bridges that were trying to exert their force. Now, if you get to this state and you lose this ADP, but there's no more ATP, the myosin stays tightly bound to actin, and that's what we call rigor mortis. Rigor mortis is when muscles stiffen after death, and they stiffen after death because you, instead of sitting in a relaxed state here with ADP and phosphate, you run out of ATP, and the muscle and the myosin cannot be detached easily from actin because it was at the end of the power stroke where they were tightly bound. So that's used forensically to help determine time of death, because if you encounter a dead body and the muscle, and you can say, oh, well, it's still kind of warm, muscles are still flaccid, probably rigor mortis has not yet set in, that puts it in a certain time window. Once rigor mortis has set in, that puts it in a different time window. And then they, forensically, they'll often talk about rigor breaking. Rigor breaks not because ATP has become available, but because the proteins have started to degrade. Myosin, actin, all the proteins inside the muscle have started to degrade because the body's been there so long. And as the proteins degrade, the muscles soften and you can start to move the limbs again. So that puts it into a different time window. Yes?
So you said that myosin doesn't necessarily, like it wants to bind to actin, but it kind of depends on where it's floating around. So does it want to bind to ATP?
Oh, yeah.
Okay.
Yeah. So I hate to give it wants and needs, since it's just a protein, but it has a high probability of binding it has an actin binding site that has a set of charged amino acids that essentially mirror the opposite charge on actin, so that when they get close to one another, they're likely to associate to one another, and then once that ionic association occurs, it exposes some hydrophobic interactions that can make the binding tighter. So myosin is sort of pre-designed to match up with actin very well and bind to actin. And a similar phenomenon occurs at the ATPase site, is that the pocket of the ATPase site where the ATP binds is just the right geometry for an ATP to fit in there, and so that's what goes in. Other things might try to go in, but they don't quite fit, and they'll come back out. But ATP does fit well. Yes?
Could you discuss all the important parts of the sarcomere?
Important parts of?
The sarcomere, just like A-band, or just like which one do you recommend?
I think it's all important, I really do. But then I'm kind of a muscle geek, so that's my problem.
Let's see if I've got the slide in this lecture or a different lecture. We can use this slide.
So when you're thinking about a sarcomere, I guess one of the first things to think about is what's a sarcomere? And a sarcomere is everything from one Z disk to the next Z disk. Anything between that space you would say is part of the same sarcomere. Now the Z disk itself is in the middle of a structure we call the I-band. So here's the end of an I-band, here's the end of an I-band. Here in blue, in the cartoon, that's the end of an I-band, that's the end of an I-band. The I-band, the definition of it is kind of interesting. It's a space between A-bands. So the A-band is where the thick filaments are, and that's in the center of the sarcomere. The I-band is from the end of the A-band in one sarcomere to the end of the A-band in the next sarcomere, or I guess you'd say the beginning of the A-band in the next sarcomere. And so it's a space that's defined by what's not there. It's defined by the fact that the myosin filaments aren't there. So that's the I-band. And the Z line is right in the middle of it. And because of this arrangement that the thin filaments go from the Z line towards the center of the sarcomere from both sides, and the thick filaments sit in the middle of the sarcomere, and they have their cross-bridge domains of the thick filaments towards their ends on both sides. When myosin and actin interact, myosin pulls the thin filaments towards the center of the sarcomere from both sides. What happens when that occurs is that the I band gets smaller. Look at the blue box in the upper diagram versus the blue box in this lower diagram, and the width of it has become smaller because the distance from the end of the A band in one sarcomere and the beginning of the A band in the next sarcomere has changed. The A bands have gotten closer together, the I band has shrunk. When muscle shortens, the I band gets smaller. The A band remains the same size because it's defined by the thick filaments, and the thick filaments don't change their size when muscle contracts. The I band shrinks because it's the distance between the A bands, and that distance will shorten as the sarcomere shortens. In this bottom cartoon, they don't even show that little blue box because there's so little space between the end of these thick filaments and the beginning of the next set of thick filaments. The M line here is the place where all of the thick filaments in one sarcomere are linked to each other laterally. That's the M line. So all of the thick filaments in one sarcomere are connected to each other at the M line, and all of the actin filaments in one half of a sarcomere are connected to the same Z line. In some, another thing I guess you could look at, I'm not planning on asking a question about this. Because there's no cross bridges in the center of the sarcomere, it looks a little different on the electron micrograph. And you can also tell from these micrographs that that area shrinks as well. It's called the H zone. So here's the H zone in the center of the sarcomere where there aren't really any cross bridges, and as the sarcomere shortens, the H zone also shortens. And that's because as the thin filaments slide into the lattice where the thick filaments are, they start to get closer and closer to the center of the sarcomere, and they start to add mass to this place that didn't have a lot of protein before. And so it gets denser there, blocks the illumination more, and you see that the H zone seems to shrink as well. So the H zone and the I band are both defined as these spaces determined by the movement of other filaments. Yes?
What does make the A band move?
In a perfect world, so what makes the A band move? In a perfect world, the A band would stay in the center of the sarcomere, and it would just pull the I bands towards the center of the sarcomere. But the world is not quite perfect. And remember, we talked about the cross bridges. Their ability to bind when the muscle is stimulated depends on their own individual position. So in theory, you should have equal forces built up on both sides and symmetrical shortening. But in practice, it doesn't always happen that way. In practice, you can sometimes get more force in one half of the sarcomere than the other half of the sarcomere, just because based on the statistical probability, you got more myosin cross bridges in one half than the other half. And I've seen these pictures before. I had a friend who was studying this particular phenomenon. The A band moves towards one side of the sarcomere. During a prolonged muscle contraction, the A band can shift towards one Z line rather than stay in the center. And that's where proteins like titin come in, because titin attaches to the ends of the thick filaments. We didn't spend much time on that here, but it's in there. It's in your textbook too. Titin attaches to the ends of the thick filaments, and it's a very springy molecule. So if the A band shifts towards one Z line, the titin on the other side gets stretched, and when the muscle relaxes, it pulls it back towards the center. So that when the next contraction starts, you start again from that position with the A-band centered. And he actually had to play with his apparatus so that he could quick freeze or quick cross-link the proteins in the muscle after a contraction so that he could catch the A-band shifted to one side or the other. If he waited after a contraction, it would always be recentered. If he did it very quickly, he could catch it while it was asymmetrically disposed, while it was pushed to one side or pulled to one side by the accumulation of force, just a bit stronger on one side than the other. So it's a real thing, and it does happen, and people have been able to measure it. And it turns out that when they did some experiments where they were able to essentially clip the titin molecules, and when they did that, after a contraction, all of the sarcomeres ended up shifted. So it seems to be pretty clear that titin plays this role to help recenter the A-band if it gets out of the center. Yeah.
So for the 4 things that affect the tension within the muscle fiber, one of them was fatigue. I was wondering if you could sort of talk about.
One of them was what?
Fatigue.
Fatigue.
Yeah.
Fatigue. So, because we didn't talk much about it in class, there won't be much about it on the exam. But it is something I think you should know. But to talk about fatigue, first let's talk about where do you get your energy from? Three ways you can produce ATP for muscle contraction. The first is you have about 5 millimolar ATP in your cytoplasm, which is used to power all sorts of things. And the fastest way to regenerate that ATP once it's used is to use creatine phosphate and the enzyme creatine kinase. Creatine phosphate will donate its phosphate to ADP, and this enzyme creatine kinase will then accomplish the chemistry so that you take the phosphate off of creatine phosphate, end up with creatine, and you now have ATP. And you have about 4 or 5 times as much creatine phosphate in your muscle than you have ATP, so that it's a ready backup supply for the ATP process. All muscles use creatine phosphate, creatine kinase to make ATP from ADP. The other ways that muscle generates ATP is either through oxidative phosphorylation or through glycolysis. And so when you're working, you use up that ATP and you use up the phosphocreatine, and when you're resting, these processes, oxidative phosphorylation and glycolysis, can be used to make ATP, and that ATP can be used by the same enzyme to put a phosphate on the creatine that's left over, so that you regenerate your reserve of ATP. You make more ATP and use some of that ATP to make creatine phosphate. And it seems weird, I don't know why the body does this, but it stores some of the phosphate on creatine phosphate and some of it goes to ATP itself. One possible mechanism, one thing is, it turns out that the oxidative phosphorylation process, and this is never something I'd ask you about, creates ATP in the mitochondria, but then it has to get out of the mitochondria. And that turns out not to be easy, but it can get creatine phosphate out. And so part of it is that creatine phosphate turns out to be a convenient shuttle to get the energy made in the mitochondria out of the mitochondria. Okay, but let's talk about how this relates to muscle metabolism. There are some muscles that use oxidative phosphorylation almost exclusively as their ATP, their big ATP engine. This is what really makes the ATP. Creatine phosphate is your short-term fix. Oxidative phosphorylation would be, this is what I need for the long haul. Oxidative phosphorylation is great for that because it can use a variety of fuels. It can use glucose, it can use fatty acids, it can use it can use amino acids that have been broken down from protein hydrolysis. It can use anything, pretty much, that you throw at it. And it makes a lot of ATP. The caveat is it needs oxygen, oxidative phosphorylation, and you have to have mitochondria. So it's a multi-enzyme pathway. It requires oxygen, takes a little time to kick in. So while the creatine phosphate is being used, the oxidative phosphorylation process is starting so that by the time you run out of creatine phosphate, you've gotten some ATP produced by the oxidative phosphorylation. And so the creatine phosphate ability to phosphorylate ADP by creatine phosphate kind of buys you some time so that you can get through this multi-enzyme pathway. When you first start to exercise, you will use glucose to fuel oxidative phosphorylation. And that glucose can come from the blood supply, or it can come from stored glycogen, which is just a polymer of glucose that your muscles can store. And you'll use that to be the fuel for the mitochondria to make ATP. As you continue to exercise for longer periods of time, you start to use fatty acids. And that's actually not a bad thing because fatty acids, pound per pound, have more energy in them than glucose. Fats have just much more density to their energy. And you can tell that by what we consider rich foods. Rich foods are the richest foods. Don't just have sugar, they have fat. Amino acids and proteins are not commonly used, but they can be used more as an emergency backup energy supply. Muscles that would use this process will be red in color. They're red in color not because the mitochondria are red, but the mitochondria are red, but because they have the protein myoglobin in the cytoplasm. Myoglobin is like hemoglobin. It is an oxygen storage molecule. And because the mitochondria require oxygen for oxidative phosphorylation, these red muscles store oxygen locally so that you don't have to worry about diffusion of oxygen from the blood supply immediately. You definitely need it from the blood supply, but you have the on-hand storehouse of oxygen inside your cell. So red muscles are red because of their myoglobin in their cytoplasm. There's another type of muscle that will use glycolysis for its energy reserves. And the glycolytic muscles only use glucose as a fuel. Again, it's a multi-step enzymatic process. It takes a little time. And so you use creatine phosphate first while the glycolysis is getting ramped up so that by the time the creatine phosphate is running down, the glycolysis is starting to provide ATP. And again, it requires either glucose from the blood supply or from those internal stores of glucose as glycogen. But it can make ATP. The downside of glycolysis is that for each glucose, you get about 2 ATP molecules. Whereas for oxidative phosphorylation, one glucose can get you 36 ATP molecules. So oxidative phosphorylation really is the champ for producing ATP from glucose. But this works, and it works well. And a good part of our musculature depends upon it. These muscles don't have myoglobin because glycolysis doesn't require oxygen. And so we call these white muscles because they don't have really a color to them on dissection. Now, for those of you who eat meat or are familiar with meat, if you think about how the meat of a chicken is divided into dark meat and light meat, the dark meat has myoglobin, the light meat does not. So the dark meat in a chicken is in the legs, in the thighs. Chickens run around a lot, but chickens don't fly a lot. And so the muscles that control their wings The pectoral muscles, the breast meat, they're white meat. Because chickens may flap their wings, they may fly short distances, but they really don't fly a lot. I guess we've bred that out of most chickens. Useful for the farmer. So dark meat, light meat, white muscle, red muscle, those tell you something about the muscle's metabolism. How does it make its ATP? So this is just a summary from your book. You can make ATP from creatine phosphate by taking ADP and adding a phosphate to it, and the byproduct is creatine. You can generate ATP from the mitochondria, which will use fatty acids, amino acids. It will also use the byproducts of glycolysis to go through the Krebs cycle and make ATP. When there is sufficient oxygen for the oxidative phosphorylation to take place, then the pyruvate that comes from glycolysis gets fed into the mitochondria and it makes ATP. When it's not, it goes from pyruvate to lactic acid, and that lactic acid has to leave the muscle and be processed elsewhere. So that's the overall summary of the way muscles will generate their ATP. And the reason why we start here is because the process of fatigue is different for different muscles. So we classify muscles based on how fast they hydrolyze ATP, how fast the myosin ATPase works. So we say we have fast muscles and we have slow muscles. But we also classify muscles based on whether they use oxidative metabolism or glycolytic metabolism. And so the slow muscles, the ones we call slow muscles, are slow oxidative muscles. Slow muscles, slow hydrolysis of ATP relative to the fast muscles. Oxidative metabolism, the oxidative metabolism must be red muscle. So slow muscles are red muscles. And they have small diameter cells because they need oxygen, and if they become very big, oxygen has to diffuse across long distances to get into the center of the cell. And so the red muscles tend to stay smaller in diameter, and they have a capillary bed that's close to the body of the cell so that oxygen can easily diffuse across the cell. As you learned in one of the very first labs, diffusion is great at short distances, not so good when you have a long distance to travel. And so red muscles stay small so that they can have efficient oxygen diffusion. The fast glycolytic muscles, white muscle, have large diameter cells because they don't care about the oxygen. They don't need it. They're using glycolysis. It doesn't require oxygen. It's anaerobic. And so they can get as big as they need to get to provide the forces that we want them to generate. And so the fast ATP using glycolytic muscles are some of the larger diameter muscle cells in the body. And the larger the diameter, the more myofibrils you can pack into the muscle cell. The more myofibrils you can pack into the muscle cell, the stronger the cell is. So if someone has large, bulky muscles, they probably have optimized their fast twitch muscle. Their exercises have been weight-bearing exercises that require force, and they have optimized fast twitch muscle. People who have a more slender musculature but who are still very active are probably more engaged in long-distance running or other events that don't bulk up the muscles. And they've optimized their slow oxidative muscles, and they get as big as they can possibly get as long as it doesn't interfere with oxygen diffusion. So it's not like those muscles aren't strong, but they'll never be as strong as the big glycolytic muscles because there are not as many myofibrils in them. Just can't because they're too small. There is a sort of hybrid type fast oxidative muscle. The ATPase of the myosin is fast. The metabolism can be oxidative, actually a combination of oxidative and glycolytic in the fast oxidative muscles. So the muscles are red, they have myoglobin, they have an intermediate diameter, and they can tolerate the intermediate diameter because they can also use glycolysis as a significant source of ATP. 3 different types of muscle. Slow oxidative, but because they're small, they generate a smaller amount of tension than the fast muscles. They're also the least likely to fatigue. So now we're getting to what your question was about. They're the least likely to fatigue. The fast oxidative, moderate tension levels, moderately likely to fatigue. The fast glycolytic, most likely to fatigue. And so if you stimulated a slow oxidative muscle continuously for an hour, and this break here just means we took a break in showing you the record, but it continues to give you the same response every time you stimulate it. And if you did the same thing for the fast oxidative glycolytic muscle, the intermediate type, you would find that it would hold on for a while, but it would eventually start to show signs of fatigue. And if you continued for 60 minutes, you would see that it's not generating the same muscle response for the same stimulus. And that's what, when a muscle cannot generate the same tension response as when it was fully rested, we say that it's starting to show signs of fatigue. The nervous system signals are the same, but the response is less. Fast glycolytic, lots of force early, but it dies away pretty quickly. It's fast fatigable. This is just the same information overlaid. So, fast muscle, more likely to fatigue, slow muscle, less likely to fatigue. It has, well, it's part biochemical, and it also points to function. If you are a marathon runner, you want to have your slow oxidative muscles optimized so that you can run for hours and have your muscles respond appropriately. If you're a power lifter, you need a lot of force for a very short amount of time. And so you can optimize your fast twitch muscles, you can lift heavy loads, but you never have to worry about holding that heavy load for a long time or moving that heavy load for a long time. So it doesn't really matter quite as much that you fatigue quickly, unless you have to do it again and again and again in the competition. So there's always a balance between the function and the fatigability. And I often say if you want to just guess what kind of muscles a person's optimized, go over to the track, the field house, and look at the different athletes who are exercising there, and you can just look at people and kind of tell based on what they're doing, whether they're running laps around the building or whether they're throwing a weight inside the building. You will see that there's a difference in their body style as well as the activities they participate in. Now I want to move along here. Oh yes, this is important. So when we try to do any activity, we use the muscles that we need. We don't recruit the fast oxidative unless we actually need them because they fatigue so quickly. And so you start off with the slow oxidative, and if you need more force, then you could recruit the fast oxidative, and if you need more force, you'd go to the fast glycolytic. Now, truth in advertising, humans don't have much of this. Most of the fast oxidative. Other animals take more advantage of the fast oxidative. Humans, it's not really a major player. We have the slow and we have the fast, but not the hybrids quite so much. But in our bodies, actually in humans, it's really interesting. Humans don't have red and white muscles segregated into different body groups. We have them intermixed throughout our body. So if you were taking anatomy, what you would find is that all the muscles kind of look like each other. They may be different in the way the muscle fibers are organized for a pectoral muscle versus a back muscle. But there's no red muscle, there's no white muscle, it's all kind of looks about the same. And that's because the white muscle fibers and the red muscle fibers are grouped into the same muscle groups in the body in humans. And so we recruit from our biceps or our quadriceps or our hamstring muscles the muscles that we need to accomplish different tasks, but we start with the slow muscles and we build to the bigger, faster, stronger muscles. Anytime you're using the fast glycolytic muscles, you've already activated the slow oxidative muscles. Factors determining muscle tension, the frequency of the action potentials, the fiber length, the diameter, how many myofibrils in that muscle cell, and fatigue. Are you tired? For number of active fibers will depend on the number of fibers per motor unit and the number of active motor units, and that's recruitment. Do you recruit just one motor unit, or do you recruit 2, or do you recruit 3, or do you recruit 10? Now, fatigue has proven to be difficult to fully understand. Many years ago, the predominant theory was the buildup of lactic acid as a result of metabolism was the reason that muscles showed fatigue. That is, as you use glucose and you continuously to build ATP, eventually so much lactic acid builds up that it causes the cells to not function properly. And it turns out that that's not true. It just doesn't work that way. It may contribute to what I would call acidification of the cell, but it's not the only contributor to that. The fact that myosin and ATPase, every time it splits phosphate off of a ATP molecule, that's called a hydrolytic cleavage. Hydrolytic cleavages produce protons. So the myosin ATPase, the calcium ATPase, the sodium potassium ATPase, all of them are releasing protons, and it's not just the buildup of lactic acid. Protons make things more acidic. And turns out that the myosin ATPase is less functional in an acidic environment. So as the acidity builds up in a cell, functionality of myosin starts to slow down in its ability to hydrolyze ATP. So that could be a contributing factor, but it's not just because of lactic acid, it's because of other acidifiers that are also working in the muscle cell too. It's not an absence of ATP. Never let anybody tell you it's an absence of ATP. If it's an absence of ATP, it's rigor mortis. Rigor mortis is dead muscle. So you exercise all afternoon, you are not out of ATP. You may have a little lower concentration, but you're not out. If you were out, your cells would die. Not out. High extracellular potassium concentration. Well, this is something that may be a contributor factor. Think about an action potential. And every time you fire an action potential, you pump sodium into the cell and potassium out of the cell. And you know that the membrane potential is dependent upon the ratio of sodium inside and outside and the ratio of potassium inside and outside. All of those action potentials can contribute to a higher net potassium concentration extracellularly, which will change the resting membrane potential and make the cells less excitable. It will get the resting membrane potential as it approaches the threshold for the sodium channels. The sodium channels will start to inactivate. That's a phenomenon that we've seen in the laboratory, that if you raise the potassium concentration extracellularly, that the sodium channels inactivate. So that makes the cells less responsive to the nerve stimulus. ADP and phosphate both inhibit the myosin ATPase. So it's called product inhibition. If you studied biochemistry, you may have talked about this process of product inhibition. That the product of the chemical reaction that the enzyme normally produces, or normally catalyzes a reaction to produce, if that product is in too high a concentration, it will inhibit the enzyme from making further metabolic transitions. So ADP and phosphate both inhibit the myosin ATPase. So that can contribute to fatigue. And the most recent contributor to fatigue is calcium signaling, that there's a dysregulation of the calcium signaling in the sarcoplasmic reticulum. So fatigue is multi-component. It has some metabolic factors like the buildup of ADP and phosphate and the acidification of the muscle cell and the potential that some of the calcium regulation may not be functioning properly. But fatigue is not just one thing. There are multiple factors in fatigue. It would be great if I could just say, you want to understand fatigue? It's this. It's not that easy. And there are still people trying to nail it down a little tighter. So for the purposes of this class, if I ask you a question about fatigue, I would probably be asking about whether or not it's the absence of ATP. I might ask a question about lactic acid, but you should know that it's not just lactic acid. I might talk about the buildup of ADP and phosphate and disruption of calcium signaling. These are the things that I would say from this slide. That's what you need to know. But in order to get there, I need to tell you about why some muscles, and so the red muscles are more fatigue resistant. Why are they more fatigue resistant? There's a couple of reasons why. Because they, and one of the most, that I lean on the most is things like the high potassium concentration, the acidification of the cell are going to affect the red muscle less because red muscle is in close contact with the blood supply through its capillary bed. And if protons build up in the cell, they can be diffused away. If lactic acid builds up in the cell, it can be put into the extracellular environment and diffuse away. If ADP and phosphate, if anything builds up, these cells have the right environment because they are close to the blood supply to dump things into the extracellular environment and let the venous system take it away. So it doesn't infect their environment quite so much. In the white muscle cells, because there's not as much flux across the membrane of these components, things tend to sit there. And therefore they fatigue a bit quicker. Also, you can certainly put into that because oxidative phosphorylation is much more efficient at making ATP, certainly if dwindling ATP supplies play a role, muscle that uses oxidative phosphorylation is going to have less of a problem with that than one that relies on glycolysis because oxidative phosphorylation is just so much more efficient at generating ATP. All right. Thank you for going there, because that was a question that I thought we should talk about with people. So this is a summary of these muscles and their different types of muscles and their properties. Slow oxidative fibers are small diameter. They make less overall force because they're small. They use oxidative phosphorylation, which requires oxygen to generate ATP. They have many mitochondria, and they're surrounded by many capillaries. They have myoglobin so that they can improve their oxygen diffusion, their resistance to fatigue, and for that reason, you will find more red muscles in places where the muscle should be contracted for long periods of time without fatiguing. So, say for example, the muscles that run up both sides of your spine, the psoas muscles and the erector spinae are mostly slow oxidative muscles. The soleus muscle of the calf is mostly a slow muscle, a slow oxidative muscle, helps you to stand up straight. And so those postural muscles are slow oxidative. Fast oxidative glycolytic, larger diameter than the slow muscles, faster myosin, so the tension development is faster. They have some myoglobin, they have some mitochondria, they can make ATP through that oxidative phosphorylation, but they also have enzymes related to glycolysis, so they can make ATP from glucose through glycolysis. They are somewhat resistant to fatigue, but they will have a larger diameter than the slow oxidative muscles, and they will have relatively fewer capillaries around them because the cells are bigger and they're not as dependent upon oxygen diffusion. And then when we add in the fast glycolytic fibers, largest diameter, more tension, fast myosin ATPase, mostly glycolytic metabolism, doesn't really depend on oxygen. Large amounts of glycogen, they will store glycogen in their cytoplasm because that's a potential fuel supply. And they'll either get glycogen from the blood supply or they'll be breaking down glycogen in their own cytoplasm. Fewer capillaries and little to no mitochondria, almost no myoglobin or no myoglobin. They can make a lot of force, but they're more prone to fatigue. And this is a table in my slides. I think it's also a table in the textbook that shows a comparison between these three different muscle types. We've got about 20 minutes left. So please, if you got questions, feel free to ask. Yes?
Can you just briefly go over like latent period, the contraction period, and relaxation?
Sure. So when a action potential occurs to stimulate a muscle cell, let's see, what happens? We went through it, well, I don't know if you were here, but we went through a description of excitation-contraction coupling. And one of the things that results from that process is that after the cell has been stimulated electrically, stuff has to happen. What kind of stuff? So you have an action potential that forms on the muscle cell membrane, and it has to move along the length of the muscle, and the movement of that action potential will trigger calcium release from the sarcoplasmic reticulum, and then the calcium has to diffuse into the cytoplasm and bind to troponin before you can have force development. So I like to think of it this way, that the stimulus for a muscle contraction is an electrical event, which is followed by chemical events, the release of calcium from the sarcoplasmic reticulum and the binding of calcium to troponin. That's a chemical event, biochemical event. You have to have release of this ion that's been sequestered in the sarcoplasmic reticulum and the binding of that ion to the regulatory proteins on the thin filament, and then the myosin has to bind to actin and it has to start its contraction cycle. So you have this biochemistry that has to happen after so you have an electrical event, and then you have this chemical event, and then you get force production. So the latent period is a period where you know that you had a stimulus, but you have not yet seen a response. right? And during that period, stuff had to happen before you could see the response. And whether you're talking about an isometric contraction or an isotonic contraction will determine how long that latent period lasts. Let's see. I know it had it open today. Ah, there it goes. So this is the slide we showed in the lab. And there's a single action potential here, and then a dashed line that's parallel to the y-axis. And these arrows here are pointing to the y-axis and the dashed line, and they've labeled the latent period. And on the x-axis, where that dashed line meets it, that's when the force development was first measured. So the latent period in an isometric contraction is that very short amount of time between the stimulus event and the beginning of force measurement. And during that period, stuff happened. You had calcium release and calcium binding to the thin filament and cross-bridge cycling. And when the cross-bridge cycling reached, here we're talking about tension in milligrams, when you started to get, maybe tenths of milligrams or hundredths of milligrams of force so that you could actually measure it, that was the duration of your latent period. And then after that, the force continued to rise. So the latent period is just that period between the beginning of the stimulus, when the stimulus occurred, and the beginning of the measurement that you want to make. Now, for an isotonic contraction, it's a little different. For an isotonic contraction, you have a stimulus that still occurs at time 0, but now we're measuring distance shortened and we're not measuring tension. So this latent period for the isotonic contraction seems to be longer. Why is the latent period longer in this isotonic contraction? The physical phenomena that are occurring are exactly the same. Exactly the same. So, is it taking any longer for the calcium release to occur? No. Is it taking any longer for calcium to bind to the thin filament? No. What's taking longer is that at the beginning of this isotonic contraction, we weren't making enough force to lift this load. You look at the isometric curve. When it first starts, the force levels are really low. If this is a heavy load, we can't move it until we make enough force to lift it. And so the cross-bridge cycling makes for a longer period of time before a measurable event occurs, makes it longer in this isotonic contraction. And that's why I've said in the lab, and I think I said in lecture, at the beginning of every isotonic contraction, there is a period where the muscle is contracting isometrically. So between 0 and the time we see movement, the muscle was contracting, it was building force or tension, but it still was not enough to move the load. And only after it generated enough tension to move the load could we measure that that load was actually moving. Every isotonic contraction starts with a period of isometric contraction, and that isometric contraction is force building until you reach whatever the load is. And once you reach that level, you can move it. And so another way of looking at that is shown, this is isometric, let's look at isotonic. Another way of looking at that is here. In this case, we are measuring the force, the tension developed. And this muscle can generate isometrically about 25 kilograms of force, but it's attached to a 20 kilogram load. And when we activate the muscle, we can see that it generated tension and it took time to get up to 20 kilograms. No movement occurred during this period. It's just sitting there until it reaches 20 kilograms. Once it gets just above 20 kilograms, then it can move. And you can't measure the movement here because you're measuring tension. But if you were measuring movement, then you would start to see the movement because you reached that level of force development where you could now move a 20 kilogram load. But it took time to get there. And that makes the latent period for the isotonic contraction longer because all the latent period is how much time before I see a response. How much time before I see a response? So if I'm measuring movement, I have to wait until it makes enough tension to move that load. So in an isometric contraction, the latent period is relatively short because you can measure tiny bits of tension. And so you can say, I saw my result of stimulation relatively quickly because you saw the tiny bits of tension. In an isotonic contraction, you say the latent period was longer because you were looking for movement and it took time to get to the point where you could actually move the load. Now, our time scale here is milliseconds, so you have to, it's not something that when in our day-to-day life we don't think about the latent period very much because we're not, the milliseconds that it occurs, that it happens for us to generate enough tension to pick up a suitcase or a backpack or whatever it is, we don't think about that, but it's there. And if you go into a laboratory type environment, you can actually figure out how long it is. And if you were looking at moving objects of different mass, you would find that the greater the mass, the longer the latent period. Because you need more time to generate enough tension to move the load.
So could you look at an isotonic, if you're looking at an isotonic, but measuring tension, would it look like the isometric?
Well, that's what this other graph was. So here is generating tension and it can't move the load. This is what the curve looks like. This is measuring tension and we can move the load. And what we find is, this is an interesting phenomenon, we find is once you cross enough tension to move the load, you don't make more tension, you just continue to move the load, but you move it as fast as you can. And that's why the speed of movement, the velocity of shortening of a muscle is also dependent on load, is because once you can move the load, the muscle will move it as fast as it can. Rather than try to generate more force, it just generates enough to move it and then moves it quickly, or as quickly as possible. So while this muscle is capable of generating 25 kilograms of force, it uses that potential energy to just move this load faster. Yes?
But what happened if it did generate more tension than that?
If it did generate more tension than this, it would, what would happen? Let me think about that. Typically that was, if you make more tension than what's required to move a load, you typically then have more inaccuracy in your movement. So think about, I'd say the best way to think about it is it's more of a recruitment phenomenon than a muscle force development phenomenon, but the same, I think the result would be the same. If there's a ball on the ground and you think it's made of styrofoam, but it's actually made of metal. Or excuse me, the other way around. You think it's made of metal, but it's made out of styrofoam. You reach in to pick it up, you recruit more muscles than you need, and your movement is not precise. It is an over movement because you had too much force. So that can happen. If you have more force than just what you need to move it, then the precision of your movement becomes less because you've got too much force to control it.
You can generate more force, it's just your body doesn't generally want you to.
We tend to recruit the muscles we need to move the load we're trying to move and no more. That's what we tend to do. You can think of, so there are a few instances where you might take you may recruit more than you need to move a load. Throwing a football long distance. You activate more muscles than you would need to just move the football in space because your plan is to launch it, or throwing a baseball. You will activate more muscles than you would actually need to move a baseball that short distance while it's in your hand, because the objective is to throw it at high velocity. So we sometimes do activate more motor units than are required for the movement, but again, it's because that's not going to stay in your hand very long. It's because you're going to launch it, or you're going to kick a soccer ball, or you do something. But again, in that case, you have also recruited motor units based on the task at hand. If you were just trying to move something, you don't need more force to move it, and so you don't recruit more motor units, you don't make more force. And so we have this internal regulation that we don't think about in terms of how much force we generate. And it tends to work like this, where you make it, if you can certainly pick up something and move it slowly. But if you're doing something, in this case, we're talking about laboratory type measurements. If we do a laboratory type measurements, what we find in an artificial environment, if we allow a muscle to lift a load, and we stimulate a muscle that's capable of lifting a load in the laboratory, it makes enough force to overcome a load, and then it's kind of self-regulated to stay at that level. I don't know if we understand that phenomenon exactly, but that's the way it works. We've got a few minutes left. There's a couple of things that I think are often asked about that we haven't talked about yet. But you guys have contributed to a pretty active session here. Let's talk about this one. If a muscle is contracting maximally and can't move a load, it's isometric. So this is shortening velocity versus load. There's no movement here. Velocity is 0. It's an isometric contraction. As the load gets lighter than that, you start to move things. Lighter goes faster, lighter goes faster. Maximum shortening velocity occurs when you have minimum load, 0 load. And this is, the relationship here is curved. It's not a linear relationship, but it is basically, you could say that the velocity of shortening is inversely proportional to load. When the load is low, velocity is high. When the load is high, velocity is low. So inversely proportional to load. In our day-to-day activity, we do this kind of contraction all the time, isotonic shortening, where we try to move our bodies or we move a load. But we also do lengthening contractions where we extend a muscle but have it still generating force. That's a lengthening contraction. The muscle's being extended while it's still generating tension. You could say the muscle's being stretched while it's generating tension. And usually those kinds of contractions have to do with the management of a load so that it doesn't fall too quickly. Sitting down in the seat, you contract the muscles in the front of your thigh as you sit down, but as you sit down, the movement of your body stretches those muscles, and it helps to regulate your descent into the chair. If you were putting down your backpack, you don't just let your arm release and drop your backpack, you control the descent to the floor. So those are lengthening contractions of the bicep when you move that load and set something down. So these are real contractions, they occur. These are real contractions, they occur. And occasionally we do the isometric case, but the isometric case probably happens the least. It's informative to muscle scientists because it's the most force a muscle can make on its own when you maximally stimulate it. It will make an isometric contraction. And the example that we often give for isometric contractions is put your palms together and press, and you can tell that you are generating tension because you can feel the forces in your arms, yet your arms don't change their position much. They're really contracting, the muscles are contracting isometrically because the arrangement that you put your skeleton in restricts their ability to shorten. So this is called the load-velocity relationship. Yeah, let's see, is it still there? And this is a version of the link-tension relationship, just a different cartoon of it. When you think about the arrangement of thick and thin filaments in the sarcomere, there is a degree of overlap of the thin filaments with the thick filaments where every cross bridge in the myosin molecule, or the myosin molecules of the thick filament, can find a binding spot on the actin thin filaments. And if all of them can contribute to the force generation, then you get maximum tension. If you stretch the muscle from that point, the more you stretch, the more you pull those cross bridges into regions where they can no longer bind to actin. And since they can't, they may have ATP or ADP and phosphate bound, but if they can't bind actin, they can't contribute to force generation. So you get less force if you hold the muscle at that length and won't let it shorten. And the more you stretch, the lower the tension goes until the point you pull the myosin filaments out of overlap with the A band, and then you get nothing. Or the I band, then you get nothing. So that is what's often called the descending side of the link tension relationship. We think of this as a little hill that we climb on this side and we descend on that side. On the other side of this relationship, when the muscle starts to get too short, what we find is that we get disruption of the order of the filaments. That is, you may end up with a situation where myosin filaments buckle because they've run into the Z lines, or the I bands, or the thin filaments of the I band meet in the center of the sarcomere and maybe dislodge each other from that symmetrical, they were almost hexagonal order that we found when we looked at the cross-section. They get offline. The distortion of the sarcomeric order of those protein filaments, when the muscle fibers get too short, when the sarcomeres get too short, causes a reduction in force production. And by now you know that that's true for both skeletal muscle and for cardiac muscle. So this box represents the regions where our muscles normally work. That is, they don't get too short and they don't get too long. And the restrictions upon the stretch, on the shortening of the muscle, has to do with how it's attached to other structures in the body, how it's attached to the skeleton, or how the cardiac myocytes are attached to each other. That provides some limit to how far those cells can stretch and how far they can contract. Some physical limits. So we work in this box, but we do sample both sides. We both have, sometimes our striated muscles will be a little shorter than optimum, and sometimes they'll be a little longer than optimum depending on what we're doing. And the best example from, on the short side, comes from the cardiac literature, where when you're at rest, you tend to stay on the short side because that gives you a little bit extra force capacity for when you become more active. When you're at rest, your sarcomeres in your heart are more on this side of the relationship. But when you get up and start to be active, the force of the blood that's returning to the heart stretches those chambers and pulls the fibers into this alignment, which allows you to have a more forceful next contraction, which then propels the blood through your body to support your activity. For the stretch side, you can think of the lengthening contractions that I've just described, like setting down your backpack or setting down a barbell. You allow, or sitting down in a chair, for example, you allow your muscles to maintain some tension while you stretch them slowly so that you can control the movement. So are we good? I would stay longer, but I have another class at 4. So, now you should know everything I've shown you, just about everything I've shown you is from our lectures, from the lab. So as you review, hopefully those slides will help. I don't know if it's actually possible. We'll see. I've tried to record this.